Well, good afternoon, everybody. First of all, I'd like to thank Alan very much for his very kind introduction. Wonderful. Thank you very much. And also, I would like to thank Alan and perhaps other co-organization for this beautiful symposium here. I feel extremely honored to be invited to Hanna Symposium, which, if I'm correct, is more than 100 years old. So I feel extremely honored. Thank you very much for this kind of invitation. Also, the title of the symposium about genome instability and evolution is, of course, close to my interest and to my mind, but I spent all of my career working on yeast. So I took the liberty to add genome instability and evolution in yeast. So if you don't mind, we will discuss of the yeast only as far for this time. When we are talking of genome instability of evolution, we of course do experiment, as you have heard, and you will hear more in this talk. And we can also look at what nature has done for us. Evolution is if you contemplate nature, you see the result of evolution. And I will start my talk by this attitude, which is rather contemplative rather than active, which for an old guy like me, it's of course, it helps a little bit because you don't have to run in your lab and do things. So a contemplation of nature started in yeast about 20 years ago when we sequenced the first yeast, Saccharomyces cerevisiae, in a large international program, including many laboratories. It was finished by 1996. 1996. The yeast genome has 13 megabase approximately, codes for 5,800 protein approximately. And at this time, we had a large press conference in Brussels to tell the novelty. It was in spring 1996. And of course, people from the press say, what did you discover? So because this thing was on the wall, but nobody understood what it is. This means simply the relationship between the different pieces of chromosome, which are horizontal here, and showing that there was some duplication in the yeast. What did you discover? And the real answer would be that, in fact, laying the basis for future discoveries is even more important than making a discovery. But we could not say that because it was not politically correct. So we were told not to say that. So instead, we answered something like that, which is blah, 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 human genome, blah, bad industry, blah, 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 blah. So it was clearly oriented toward applications. At the time, human genome was not known by industry. We were unable to analyze the sequence, and gene function, of course, we were starting to study. So in fact, this has changed completely the field. It was a starting point for the field, and because at the time there were a few surprises. And these surprises, 20 years later, we now understand them, and they have changed the way we view genomes, they have changed the way we view biology a little bit. Because we find, first of all, several orphan genes. Genes have no homologue anywhere. And of course, their origin and nature was very mysterious. And this is true until today. All genomes have genes without homologues anywhere else. Our genomes have genes without homologues anywhere else, because in fact it reflects a creation, the dynamics of creations of genes, which is very intensive. And we also find something, there were many paralogs, and this was in apparent contradiction to basic neo-Darwinian idea. Why don't you want to have two genes to do the same thing if only one is necessary? And this figure shows that in fact there are many cases of duplication in the yeast, and this is again a general phenomenon. So far, we don't know a single yeast genome which is minimal. Nature never creates minimal genome. Nature always creates redundancy, because in all of this garbage, this is how you succeed in evolutionary. And then we discovered a little bit later that only a small proportion of the genes were essential for life. And this is also very critical, because if you believe that genes are essential for life, the consequence is that all mutation will be deleterious, or most mutation will be deleterious. If you believe that most genes are not essential for life, you can create novelty which will be positive. So I will discuss a little bit along these lines. First of all, the first, very early after the sequence, the first idea about yeast came from two pieces of work, one from Wolf, in which he showed, by informatics comparison, he showed that the yeast cerevisiae genome, Saccharomyces cerevisiae genome, was in fact the result of an ancestral complete genome duplication followed by a very important gene loss. 90% of the genes that were duplicated get lost subsequently, but the genome shares the traces of that genome duplication. And the second surprise that we had a few years later was we wanted to examine evolution as it is. So at the time, we had several yeast species, of course, but we did not know how they compare. They look very similar in terms of physiology, biology, and the like, but how were they in terms of genomes? So with the means of the time, we sequence, you will look at this number, 5,000 reads per species. Today, we produce millions of reads every day in the Illumina sequencing. But at the time, it was Sanger sequencing. We produced 5,000 reads. On these species, they were selected more or less, with a thumb, because we did not know very much of the phylogeny. And what we discovered, based on amino acid identity or based on synteny of genes, we discovered that the yeast were extremely large in terms of evolutionary distance. The two closest yeast species are not very close. There is no such thing as two close yeast species. Today, we know much more. In the book of Kurtzman, recently published, he lists about 1,500 yeast species. About 2/3 of them are ascomycete, another third are basidiomycetes. The budding yeast, of course, is the largest group. The budding yeast is more than half of all yeast. So Saccharomyces cerevisiae and others become the model because this is a single monophyletic group, and I will speak mostly of this group because this is where we have the data. In addition to that, there is another group of yeast, and we just had a talk on Pombe, but there are few yeast from this group, and there are yeast in the basidiomycete group, mostly in these two basidiomycete subgroups, Puccinio-Mycotina, Agarico-Mycotina. So yeast are not monophyletic. At this point, we believe that yeast are the evolved descendant of preliminary filamentous fungi, which is important to have in mind because this means that the evolution of yeast is mostly by a loss of something, loss of function, loss of something, which is probably a very important signature in terms of evolution. Evolution is usually going downwards, except for specific cases where it is, of course, creative of novelties. Okay, in terms of genome, the bias in favor of the budding yeast is even larger. This is a picture I put almost a year ago, which is slightly out of date. But you see that we know a lot of bad budding yeast, very few bad Basidiomycotina, and of course the fission yeast, which are here. So most of the things I will discuss about the evolution will be about the budding yeast because this is where the data are today. This is the status of the knowledge of genomics of budding yeast. Relatively recent, this year. We now consider that there are four different, among the budding yeast, the monophyletic lineage, there are four different genomic architecture. The genomic architecture typical of the family of Saccharomyces cerevisiae, and this is the list of species which are sequenced. For some of them, we have dozens of isolate sequence. For Cerevisiae, we have thousands of isolate sequence. But this is a list of species sequence. There is a group of Saccharomyces cerevisiae. Half of them have inherited the duplication. Half of them are pre-duplication. We have another group that we collectively call CTG because they use a different genetic code, the CUG codon. It's coding for a serine rather than a leucine, and this is of course a major change, and dozens of thousands of codons have been also replaced in their genome correspondingly with this change. This is a number of species. We discovered recently a third group, which is, we have less. We call them methylotroph. The reason there is no family name is because the taxonomy do not fit exactly the phylogeny. And there is a basal lineage represented so far by the Yarovia group, and of course the Schizosaccharomyces, which is outside. If you look at the anatomy of this genome, it makes sense. The classification is perfectly consistent. The classification is consistent with protein-protein comparison. It's also consistent with the anatomy. The Saccharomycotina yeast, or the budding yeast, have very few spliceosomal introns, whereas the Pombe yeast has many spliceosomal introns. So they sort of probably lost the machinery of loss of the intron and the RNA machinery very early in the evolution. They have a very compact genome with high coding density, over 70% protein coding, compared to our genome, which is less than 2% protein coding, except for Yarovia, which is less. This is something that very few people discuss, but the way they use genetic code, I told you that this one used genetic code by changing the CUG codon, but these use the genetic code with using tRNA, which are equivalent to the bacterial way of reading the genetic code. These use the genetic code like normal eukaryote. This is why we suspect they are probably ancestral. And this is a mixture, intermediate. Some of them use bacterial, some of them use eukaryotic way of reading the genetic code. In addition to that, this Saccharomyces group have the point centromeres, where others have large centromeres. They have the triplication of the mating cassette that Jim was talking about, where others have not. Pombe has also a triplication of genetic cassette, but this is for a different reason. It's an independent, it's a converging evolution. And something which is not very frequently mentioned as well is that all of these yeasts have complete mitochondrial DNA, in particular with complex 1 coding, but not the Saccharomyces and the group of Saccharomyces, and not Pombe. And this is again a convergent evolution, the loss of complex 1. Okay. Based on that, we could attempt to recapitulate the evolution by putting a change genetic code somewhere here, okay? Switch to bacterial decoding mode somewhere here, and here the invention of point centromere, mat triplication, loss of mitochondrial DNA genes, and so forth, okay? Each of these subgroups here itself is extremely diverse. We have enough information to tell for a new genome sequence, if somebody sequences a new yeast, we can tell whether it will belong to this group or this group or this group. No problem about that because the signatures are strong. But within each group, we have a lot of divergence. If we compare, for example, average amino acid identity, or if we compare the conserved synteny, the proportion that the genes are next to each other in a conserved fashion, you will see that intergroup comparison show a low proportion of amino acid identity, as you might expect, as well as a low synteny. The intragroup comparison shows slightly more amino acid conservation, as you might expect, but still higher synteny as well. But if you look at the number here, you find a divergence which are of the same order of magnitude as if you look at the vertebrate from one to another. Okay, and this is an illustration of this is two yeast from the CTG group, so they are clearly compared to each other. If you see the synteny already is largely disappeared. Same conclusion if you compare the proteins. This is within sorry, this is between three groups. One example of Saccharomyces assay, Saccharomyces cerevisiae, one example of CTG, the Baryomyces, one example of Basel group. And you see that the number of proteins which are common to all three is only 40% of the total number of proteins which are in the three species. If you do it within group, you see a significantly increased fraction of core protein, roughly double, 77% here. This is by comparing three pre-duplication Saccharomyces assay or post-duplication Saccharomyces assay. You see about the same figure, about 80%, but still 20% of genes are present or absent in one species or another, which goes with the same idea. There are orphans here and there. So at this point, we conclude that evolution of yeast is a constant circle between gains and losses. Losses are very important. They occur by single gene deletions or by pseudogene formation, but mostly by single gene deletion. I will illustrate that in a second. Gains occur by a variety of mechanisms, which includes the de novo gene creation, which for some time was not considered very probable because creating a gene out of nothing is not the most easy way to do, but in fact we have examples like that, as well as by horizontal acquisition. I will illustrate that a little bit. The total number of genes which varies within limits which are not large, so therefore the gains and losses on a large evolutionary timescale are morally compensating for each other. This is just to illustrate the loss of gene. Okay? We've recently compared two Baryomyces species. The Baryomyces Thyrocola onseli. They are 99% synthetic. They are close enough to be synthetic. So we could screen, this is ACT screening, we could screen what you see here. That gene or two genes here are present in this one. This disappeared from this one. These three genes here are present in this one, disappeared from the other very easily, okay? And we have checked that these genes are not transposed anywhere else. They simply disappeared from the chromosome, okay? And if we recapitulate that for the entire species we have studied, starting from onseni, which considered as a reference, Thyrocola has 90% sequence nucleotide identity, 5% divergence. Remember that we differ from chimpanzee by 1.5%, okay? That's 3 times more. Fabry, 15% sequence divergence. During this evolutionary time of sequence divergence, we have lost 11 genes on this branch, 9 genes on this branch, and 106 genes on this branch. In a different pairs of yeast, taking this one as reference, 12% sequence divergence, we have lost roughly 100 genes on each branch. So as a clock, if you want to have a clock of evolutionary loss, this will be 2-3 genes are lost for a nucleotide divergence of 10%, which is not rapid. Okay, it's not. You don't lose all your genes at every generation, but it is significant on evolutionary time scale compared to the number of division of the yeast. Now the gain. The gain are gene gain are not gene number gain is made by duplication, but gene species gain is not made by duplication because duplication creates twice the same. This is just to illustrate that in fact in every yeast species sequence so far, it is an old slide from the first one we had, about 30 to 50% of the genes are not unique. So the fact that there is no minimal genome is a very important lesson to take home. Our genome is not minimal. There is no minimal genome. Evolution is nature, does not think the same way as an engineer would create a novel genome. It's a very different way of thinking. Because the genes are constantly duplicated and constantly lost. And the next way to gain gene is horizontal gene acquisition. And this is a slide in which is already a few years old, out of date, but just to illustrate the point. This is a gene we have identified in different yeast, shown here, name of the yeast shown here. And this is the gene we have identified, and the function, the predicted function for most of them, the function has been demonstrated for a few of them in S. cerevisiae, such as uracil synthesis or so forth. But for most of the time, the function is assumed according to the function described in the suspected donor of the. So a number of genes, and in every yeast, we have genes of bacterial origin. In every yeast, mostly they are of different sorts. We have a few genes conserved in the phylogenetic branches, but not so many. So we have acquisition of this bacterial gene. And in fact, this is probably very important to create the novel lineages, because this brings different properties, different functions. Most of them, if you look carefully, are metabolic enzyme here, but not all. Okay. Of course, we don't know how the DNA gets from bacteria to yeast, but we suspect that it goes by simple transformation, because if you look at the size distribution of those genes, they are significantly smaller than the size distribution of the other genes, which suspect that, in fact, the shorter DNA transfect more than large DNA. Okay. Not only that, but we find not only small pieces of DNA, but we find huge pieces of DNA introgressed into yeast. And this is something we recently discovered in Cureicia capsulata, a member of the methylotrophic group. In this piece of the chromosome, there is 5 genes, 3 of which, in order, comes from a nitrate reductase cluster. So we have the transporter, we have the nitrate reductase, and we have the nitrite reductase. The yeast are not supposed to use nitrate, but in fact, this group of yeast, this one and the others, use nitrate. And in fact, having captured a piece of DNA, with in this case 3 genes from plants or from other sources, gives them a novel phenotype, which is ability to use nitrate as a nitrogen source, which is of course different from the other. When we look at this group here, you see this cluster in all the species, except this one, which is the outgroup here. We do not have the cluster. So the transfer has occurred somewhere here, somewhere on this branch here, based on the parsimony thinking. So this is a cluster of genes, probably of plant origin, which has changed completely a lineage of yeast. In addition to this long distance introgression, there is one here, there's another one described here, between different yeast and a wine yeast in cerevisiae. In addition to that, people have published for years that we can make hybrids between all of these yeast, and the hybrids are usually alive. Okay? They are not necessarily fertile, but they are usually alive. But this is artificial hybrids published by experiment. In addition to that, we know now more and more that natural hybrids exist in the yeast, and they were probably overlooked in the past. We have a collection of hybrids in the Saccharomyces group here. These species are all sequenced. They diverge tremendously in nucleotide. Uvarum diverged from cerevisiae by 33% of its nucleotide, but they form a hybrid, either haploid-haploid or diploid-diploid, or also haploid-diploid. We have the 3, and they are perfectly alive, and they probably contribute to evolution for a reason I will show in a minute. We have hybrid in this group as well. We have discovered hybrid in this group, discovered hybrid in this group, and in fact, they are hybrid as well in the Basidiomycota yeast. So it seems to be a general phenomenon. So the evolution of the yeast is not only a tree-like, but it's a network-like. They exchange their complete genome. So just to conclude about that, all of the experiments have shown that, in fact, the incompatibilities predicted by Dobzhansky-Müller have not extremely frequently occurred, at least the dominant one, and they are mostly nucleomitochondrial. In fact, hybrids were disregarded for evolution because most of them are meiotically unfertile. But there are mechanisms to restore fertility in the yeast. One of these mechanisms is by doubling the genome. So if you double the genome, you make a tetraploid out of a diploid, you immediately restore the meiotic fertility. So it's creating a way for evolution to start out of hybrid. And then there is a mechanism that we discovered in one of these hybrids, is that they evolve by, some of them by loss of chromosome, but mostly by loss of heterozygosity, creating chimera. This is why I have the Babylon monster here, okay? One of these Babylon monsters we discovered was Millerosima sorbitophila. It has 7 pairs of chromosomes, very nice. It looks like a diploid. This pair of chromosomes is strictly heterozygous, differs by 15% nucleotide substitution between the two parents. This pair is strictly homozygous. This pair is strictly homozygous. This one is heterozygous. And those here, if you look carefully, they are heterozygous on one side and they are homozygous on the other side. So they look as if there was a loss of heterozygosity process which goes on in the evolution of this hybrid, which will eventually create homozygous cell line of different combinations. So they are chimera of their two ancestral parents. The same figure was observed recently for a hybrid of Saccharomyces, a different group. Very much the same figure, homozygous piece here, homozygous piece, or in another hybrid from the same group. So for those of you who are geneticists, this is a very important take-home lesson, because a hybrid which is heterozygous, a population of heterozygous hybrid, you learn in the genetic course that they will mitotically give birth to identical clones, but they don't. They give birth to heterogeneous populations of chimeric genome, which are partly homozygous, different parts. If you think a little bit, this is very important. If you're a microorganism, what you don't want is your progeny look like yourself. You want a progeny which is as diverse as possible. And this is probably one of the mechanisms the yeast use a lot. They make hybrids because they mate with everything. So they don't care, okay? Most of the hybrids do not survive, who cares? They make hybrids, and they solve this problem by loss of heterozygosity. They create large populations of different progenies, out of which some of them are successful, and this is the one we see, okay? So this brings me to my second part of my talk, which will be going back to the lab and do some experiment. Okay? And this is just a slide I put for students. What I'm going to argue here is that compared to the nucleotide changes, which is of course one source of evolution, compared to the nucleotide changes, the chromosome dynamics is more important. Okay, and if you make a simple calculation, these two guys, very close siblings, they differ by 1.3% of total in terms of SNP or indels, okay? But we differ by over 1,500 inversions. If you calculate how many nucleotides are in this inversion, this is 5% of total. So we differ from chimpanzee by 5% of our genome, which is not oriented the same way in the chromosome, and only by 1-something%, which is based by nucleotide substitution. And if you look in the literature, either in natural population or after experiment, segmental duplication have been mentioned, and they are very frequent. And even in cancer, multiple rearrangement, this was already mentioned, I think, by Jim, multiple rearrangement occurs. So the chromosomal rearrangement is probably very, despite the fact that there is single point mutation, but in addition to that, chromosome rearrangement is probably very important. Today, if you look, compile the literature, we have amplification in yeast cerevisiae, which are of the type of segmental duplication, usually large segment, either interchromosomal, very much like our genome. We have this is new compared to our genome. We have new chromosomes. So simultaneous duplication of chromosome pieces create a new chromosome. I call them neochromosomes. But so I have strains of yeast with 17 chromosomes instead of 16. So I will call them Saccharomyces dujonensis just for fun. Okay? And we have subtelomeric amplifications. And of course, we have episomes of various sorts, usually centromere less, some with centromere but very short, and palindromic center. This corresponds to low-order amplification, usually duplications, sometimes up to 3, 4, 5, up to 5 copies, but no more. Okay? And the literature about evolution in Cerevisiae is plentiful. This is just a list of papers, not complete, I have collected so far. But most people ask the same question. If I take a wild-type yeast, put it in limiting conditions or detrimental conditions, what happens? So if you put yeast in glucose limitation, sulfur limited, phosphate limited, nitrogen limited media, etc., you find a lot of mutations. And people are happy to find, but they are, the answer is in the question. If you put them in glucose limited medium, they amplify glucose transporter. If you put them in sulfur limited medium, they mutate in the regulation of sulfur and so forth. So in fact, this type of experiment has been done, and the results are not so surprising. We and others have done a slightly different, it was already mentioned, with gene dosage, gene balance, so it's ribosomal protein. And we, in this case, the yeast genome is not optimal because they are missing some ribosomal protein or histone in the case of histone, and they produce duplication of the remaining genes. So I was interested in something else. If you think of cancer, cancer develop not in a limiting environment. They develop in your body. The body is good environment, okay? But the cells are genetically altered. So I was thinking that what would be the spontaneous evolution if I put yeast in a most favorable environmental conditions? I put rich medium, appropriate temperature, and the like, okay? But I take, of course, irreversibly altered genome. So they are very sick intrinsically, but there is no selection from the medium. This is the idea, okay? So then we started a series of experiments a few years ago. It took us some time because we were looking for genes to mutate, which will be essential. Of course, that's not so easy. And they will be irreversibly altered. So we are looking for deletions of essential genes, which is not, of course, feasible. So instead, we were looking for replacement of essential genes by genes from a very distant yeast, such that the proteins will be so different that they will hate it. And in fact, they do. Most of the time, the experiment failed. The cells dead, simply. We succeeded with the amino acid tRNA synthetase, the asparagine amino acid tRNA synthetase on chromosome 8, about 67% sequence identity with the one of Yarovia. Those are large genes, 1,500 nucleotides. Same for lysine tRNA synthetase on chromosome 4. Okay? Each tRNA synthetase charged cognate tRNA. The tRNA differ between Yarovia and Cerevisiae, and each tRNA reads quite a large number of codons, of 200,000 right here. Okay? So we did that. And in fact, we did it in diploid. We made tetrad analysis. And what you can see here, that the yeast are very sick. They grow very poorly. This is a rich medium. This is a normal Cerevisiae gene. This is the duplication time, 100 minutes. The duplication time of those guys with the Yarovia gene, about 400 minutes. Okay, so they are extremely sick. Okay, haploid or diploid, but they survive, and they do mating, they do meiosis. We can do genetic with them. First thing we are looking at is why are they so sick? Okay? Why are they so sick? And we concluded they are extremely sick because, as you might expect, there is a low frequency of charging of the cognate tRNA. And the way we showed that is if you look at the complete transcriptome of this mutant compared to a normal cell, you see they are grown on rich medium, and you see that the amino acid biosynthetic pathway, which are described here, the genes are always overexpressed. So on rich medium, the genes should be turned off because the yeast does not synthesize amino acid on rich medium. But in this particular construct, they are turned on very significantly, which indicate by the GCN2, GCN4 pathway, which indicate that there are lack of tRNA, charged tRNA. So we took these strains, very sick, and we grew them for 200 generations. At the beginning, it was very slow because they go with 400 minutes division instead of 100, and then they accelerate over time on rich medium. I should insist on that. And there was no bottleneck or very limited bottleneck. This is not subcloning. Okay? This is just taking a population of 1 billion cells, transferring that to the next medium. And what you can see here, original strain were small, and we have larger colony which appeared and eventually invade the population. We sequence them all, put them on pulse field gel analysis, sporulation, and so forth. And what we discovered, to our surprise at this time, was that all of the mutants we have analyzed, which were growing much faster than the original strain, there were a large number of amplifications, segments of, in this case, on chromosome 8, 30, 50 kilobase. We find them in up to 14 copies. And if we run the chromosome on pulse field gel, you see that the normal size chromosome would be here. And we have, in addition, a larger size chromosome, which is here. This is made on diploid cells. This is where we have 2 copies of that. If you make a simple mathematics calculation, this, if you multiply 12 additional copies by the size of the copy, you predict 943 kilobase, and you observe this. So we concluded that chromosomes have expanded almost double by amplification of an internal segment on chromosome 8. That internal segment carries asparagine from Yarovia, asparagine synthesis from Yarovia. On chromosome 4, the same lysine. Very much the same thing, a little bit more complicated because it's larger, but we have amplification of larger fragments, 50 to 200 kilobase, up to 24 copies. And again, we have chromosomes, normal size is 1500 kilobase. The new chromosomes are 2500 kilobase. Okay, so we call them macrotein because I was looking for a word which did not exist yet in the literature. We have verified that besides this amplification, the rest of the chromosome was perfect by complete sequencing and so forth. And if we zoom on the limit of the amplified segment, we find something interesting. The amplified segment here on the right border, we have sequenced it, so we know exactly where it is, follows in a LTR of a solo LTR from transposon, but this is a sigma family, a TY3 family. The left border fall into a TY4 LTR element. The two have absolutely no sequence identity. And in fact, there are internal border here which fall within 2 solo delta, which share 70% identity in this case. If you do the same thing on chromosome 4, you will find a piece end here falling in an intergene, which has absolutely no sequence identity, with the other end which follow delta here. Okay. Out of that, and looking at PCR sequencing junction, it took us some time, we eventually determined the structure of these amplified chromosomes. And we find in every experiment we've done, we find 2 things: formation of very large circular episomes and formation of the macrotein chromosome as shown here. But in the 2 different experiments, first one we had circles first, and then macrotein later on in evolution. Second experiment, we had the macrotein first and the circle afterwards, which suggests us strongly that in fact the two phenomena are separated from one another. It can go one way or the other. They are not directly dependent. If we look carefully, I think this slide was shown by Jim already. If you look carefully at the sequence border of these amplicons, which are here, you see here that there is a crossover, if you want to call it that way, but involving a single nucleotide sequence homology, which is probably, I don't know if Jim will agree, it's probably too small for classical crossover. So there is a junction here, we have sequenced a junction between sigma 2 and tau 2. And it occurs exactly as shown here. Okay, so the amplification limits are non-homologous regions. In addition to that, in the macrotein chromosome 8, there was a deletion, as you might expect, because the centromere is here. So you cannot amplify the centromere on the same chromosome. And in fact, there is a deletion between those two delta elements, and they share this sequence. And the crossover here occurred within this sequence, forming a repeat unit made of this sequence plus this sequence joined together and repeated 12 times, so 12 additional times along the chromosome in tandem at the right place, because we could not, we've mapped it exactly, and this is simply the original piece of DNA, which was here, which has been tandemly amplified along the chromosome. So the result is, as shown here, 12 additional copies, 30 kb repeat unit with either one no homology or limited sequence homology. For the other macrotein on chromosome 4, very much the same thing happened, except that the repeat unit is flanked by a quasi-palindromic junction formed in the intergene here, without any sequence homology in this area. So it's a replication hairpin, basically, which occurs in this area. This is one end, and the other end is between two solo delta, which are opposite ends, such that, in fact, we have a palindromic repeat, 22 copy of inverted repeat of the amplicon. Okay. As you might expect, genes are over-amplified to some extent. We verified that by running again transcriptome analysis, comparing the mutant, the amplified mutant versus the parental strain. And you see that on chromosome 8, genes are over-amplified roughly 6.2 times on average. And the ratio of copy number is 7, so more or less the same. Same on chromosome 4, genes are over-amplified 16 times, the ratio is 12. So the genes carried by the amplicon are over-amplified approximately as you might expect from the number of copies which are in these cells. So these cells should be imperfect. They should not be optimized actually, but in fact they grow better than their parents, okay? So at this point, we were asking, how do macrotein chromosomes arise? Are they stable? What is the basis for the phenotypic recovery? And of course, everybody has, in this field, everybody thinks this way. Segmental duplication makes 2 copies. Unequal crossover makes 1 and 3. And then strong phenotypic selection makes many. And this is exactly what happened in the amplification of Plasmodium when you select for resistance to inhibitor of DHODAs as published here. But we could not find anything that fits with that because, in fact, we have either 1 copy or many copies, but we have never found any intermediate form. So we suspect that it goes in one step from 1 copy to many copies by the sort of rolling circle type of mechanisms. The way we've shown that, because the referee did not agree on our interpretation, we have analyzed and sequenced 20 or so isolates from the first evolution experiment, same for the second evolution experiment. We find either circle or macrotein for this one. We find either macrotein or circle for this one, but we could not find any intermediate about 20 isolates. So therefore, we had to conclude about that. The second aspect is the number of copies of the exogenous synthetase, in absence of the Cerevisiae synthetase, number of copies of exogenous synthetase, of course, is selected for when you increase from 2 to 8, but not much if you increase further, or 3 to 6, and not much if you increase further. The duplication time of these two is more or less the same, despite the fact that you have 4 times more copy. Okay? We have then retransformed those strains by plasmid-bearing Cerevisiae gene or Yarovia gene, and again you can see between 4 and 13 copy, or 3 and 10, or 3 and 12, more or less the same growth rate for this. And we have finally check whether there were suppressor mutations, because we have the complete genome sequences. We finally check whether the suppressor mutation, this is published as such. The conclusion is that we find a few heterozygous mutations in variety of genes, as you might expect for 200 generations under the neutral hypothesis. But in fact, we do not find any mutation which could have been selected for of any sort. So at this point, we had a model which is we suspected that, in fact, the way to create this macrotein chromosome is a template switching during the replication. If the template switching occurs in this topology or in this topology, and if switch occurs like this or like this, and if the second next door origin fires during this way, during this time, then it may use this DNA as a matrix to create the rolling circle type of amplification, as either direct tandem or inverted tandem, as we have seen here. Okay, that's our model. These macrotein chromosomes appear extremely stable compared to what you would expect, because you have 20 copies of same thing in tandem, so they should be, and they are extremely stable. Again, we grew them for more than 150 generations. We analyzed them, and they 4 out of 4, 2 out of 2 were identical. We transformed them with plasmid with the Cerevisiae gene. We grew them again 150 generations. 2 out of 2 were unchanged. And in this case, we had 1 out of 2 which was unchanged, and 2 in which the macrotein has disappeared. So eventually, they will become unstable, but it takes time. So summary at this point is that we have junction between non-identical sequences forming a tandem repeat, or forming inverted repeat at this point. We published this paper, as Jim already showed, and concluding that this type of rolling circle type of amplification is probably triggered by the stress that we impose upon. We then ask the question about what happens. Everything has been done on diploid, which is a normal way of living of Saccharomyces cerevisiae. They are naturally diploid. They are on the lab, they are haploid. And what would be the result if we work with haploid cells? So we were experienced with the previous work, and we knew how quick it was to create mutations. So we reconstructed haploid cells times 0 by tetrad. And you see they are extremely sick. We put them on minimal number of generations. And then we subclone them, okay? And some of the subclone, we run the evolution experiment and do the same thing, okay? What happens? First experiment we found, this is haploid, okay? We find instead of, we find the normal chromosome plus a new macrotein chromosome, double the size of the normal chromosome. They were all identical, okay? So something had happened which makes disomic cells, because we verified again by sequencing the other chromosome, we are not talking about disomic cells. One of them was huge macrotein chromosome. When we look at the other experiment, we found that, they were all disomic, but whereas this one have a normal chromosome plus a large chromosome, here the normal chromosome has disappeared and they have 2 macrotein chromosomes. At this point, we saw that, this is the size, at this point we saw that and we put it on the shelf for 6 months until we had more inspiration of what to do, okay? Because the two experiments seems to give completely different results. Then after some months of thinking, we did the same thing. Sequencing, we identified amplicon, but low frequency of amplification, small number of copies, okay? If you look at these amplicons in details, you will see that all of them end in the CUP1 region of chromosome 8, okay? The CUP1 region of chromosome 8, we measure the number of copies again by sequencing. Normal number is 16, okay? And in the mutant we have 200, 500, up to 1300 of CUP1 amplification. So we had simultaneously low-level amplification of large pieces of chromosome and extremely high-level amplification of the CUP1, okay? It turned out that the CUP1 locus in Cerevisiae has no EcoR1, no BAM1 site. So if we digest this enzyme on pulse gel, we can measure the size of the novel CUP loci, 16 copies for the wild type, and so many copies for the other, and we can determine how many copies we have of the amplified CUP locus. So this was very, very strange, okay? So we had expansion of tandem array, and if we look very carefully in the CUP locus, in each of them, we find one and a single one quasi-palindromic junction. And this is probably something which will be important for the conclusion. Whoops. We find in this particular case here, we sequence it and we find a junction like this within the CUP1 repeat in one copy, and there is 110 repeats in total. In addition to that, we find a junction between two delta elements such that we could propose for the structure normal chromosome plus a macrotein chromosome with one amplified CUP locus and two normal CUP locus shown like here. Same thing, and I will go quickly on that because it's again the same idea. But basically what we have is a quasi-palindromic junction, not the same, in the CUP locus for this mutant. Here we have a junction between the intergene, which is at the end of this fragment, and a CA repeat of telomeres, which allowed us to propose a structure in which we have two macrotein chromosomes with two amplified CUP loci flanked by segmental duplication, one of which is attached to a novel telomere. And finally, the third type, yet another quasi-palindromic structure in the CUP locus. And again, amplification as well. The same structure, two amplified CUP locus flanked by segmental duplication, attached this time to a chromosome 16 piece of chromosome to make the centromere. So at this point, of course, you are completely lost with this structure. I can understand that because it looks like a mess, okay? But they are, if you are a yeast, they grow happily, where their parents were not growing happily, okay? At this time, I think we have a model which explains everything and which may be of interest, and I will stop on that. The model is the following. Here's a chromosome 8, and like we have in our chromosome, we have repeated sequences, delta elements. They are not identical, but they are sufficiently identical to provide minimal sequence homology, okay? We postulated the fact that, in fact, interaction between those two first create a disomy, and create in this disomy one of the two chromosomes is almost entirely duplicated because the interaction between that and that duplicates the whole central part, which is shown here. This is under bracket because this was a hypothesis. I will show you that we have data for that. Now, if you have a chromosome like that, if you inherited a chromosome like that, several things may happen. And we next postulate, and we have based on our experiment, is that if you take this, there are three, in this structure, there are three normal CUP loci, okay, which are themselves 16 copies of CUP loci. If you do hairpin replication in this one here on the left, you will end up by amplifying the copy of CUP1. At the same time, you will amplify the left flanking amplicon, which is here, which of course creates an invariable structure. But if this, at the delta element here, this amplicon may interfere with this one and create the structure we have seen. Of course, we have this hypothesis because we have seen this structure. If you make this model, you will, this is the one we have here. If you make this model, same thing may happen here. Exactly the same thing. It will duplicate the next piece of sequence along the chromosome. And because it cannot interact intramolecularly, because there is no delta anymore on this side, it will interact intermolecularly with the telomere of another chromosome. We think this is correct because we have found the intermediate among the subclone that we have isolated. We do have parents with one normal chromosome, one macrotein chromosome, in which there is no amplification of the cup locus. So the amplification of the cup locus occurs afterwards, which means that if we have that, how do you explain the formation of two macrotein chromosomes out of one? We suspect there is a subsequent interaction between those two guys forming what we have observed here. The conclusion for that is that, in fact, what we suspect is accidental template formation within a cup1 repeat, during replication probably, creates the high-level tandem amplification of rolling circle type of the flanking sequences. Segmental duplication of the flanking region, which is then resolved by interference between dispersed repeated elements. So this is the idea that simple accidental events at the replication fork and the appropriate chromosome structure may have absolutely dramatic consequences for the offspring. We submitted this paper recently to eLife. They was immediately rejected, based on the idea that we were doing artificial experiment and there's nothing natural. To my understanding, an experiment is non-natural by definition, but anyway. Then my colleagues, Liti and Shashreer, have a program of sequencing 1,026 strains of Cerevisiae already sequenced from the wild. Okay, this you can find this here. These sequences are already done because I asked them. They look at the cup locus and they find a number of cup locus. Most of the strains have 16 copies, as you might expect. Some have slightly more, and we find a dozen of strains here which have more than 300 or 400 copies of cup. So they are naturally exactly the same as the one we have artificially created. I don't think I will send it back to the same journal, but if you have some suggestion, I can look at that. Okay. So at this point, I will stop. We have created this type of structure, very complex, very abnormal, very unnatural structure, starting from genetically altered genome grown under optimal conditions. The phenotypic recovery is obtained by massive chromosome rearrangement with extensive amplification, which are themselves not directly selected by something, which is some similarity with the cancer. And the arrangement includes the formation of complex chromosome, and we suspect interference between replication bubble. At this time, I will stop. This is over the years, we have collaborated with a number of labs under the Genome Consortium and the extension now to the larger group of people, collaboration with the sequencing center at Genoscope. My lab is now formally closed because I'm retiring, but it's now formally closed. People are already left or they will be leaving. This is the people who were there a few years, only 2 years ago or 3 years ago. The experimental work was done by informatics work by Baun Kada, experimental work by Agnès Thierry. So I thank you very much for your attention. Yes.
Did you also check for the activity of the enzyme itself? So the idea is here that you compensate for the charging activity by increasing the copy number.
Yes.
But did the enzyme evolve as well?
No, we have not done enzymology, but the enzyme did not evolve because we sequenced it and it kept the original sequence. In addition, if you look at the numbers, there are 550 codons approximately. 1/3 of them are different. So if you want to change the enzyme, you want to change many things simultaneously. So there is no way to change it. And we have sequenced them, and they are absolutely identical to the original sequence. But we have not looked at experimentally by enzymology of the charging. Yeah.
And the tRNA also did it?
No, the same for the tRNA. That did. Yeah.
So am I correct that there are transfers of DNA sequence from bacteria to yeast species that do not use the exact same genetic code?
Yes, there is. Absolutely, there is.
And so, do you know that those genes are then functional in the yeast?
Not for all of them, but what we frequently observe is that they start to duplicate in the host cells, so which suggests that they will do something. But I don't think it has been demonstrated in this CTG group. This is a serine CUG codon, okay, which is not one of the most frequent leucine codon in bacteria. So it may be that by chance there is no CUG codon in some of these genes. I have to look carefully on that, okay? But it's not a frequent codon in the bacteria.
And have you observed any yeast to yeast horizontal gene transfer?
That's a good question. It's not that easy to do because if you look at the phylogeny of the yeast, you don't have beautiful trees. The bootstrap numbers are terrible usually, probably because they do a lot of these exchanges, and some of the genes have different origins. So it's not that easy to find a long distance transfer from yeast to yeast. We have long pieces of DNA from Zygosaccharomyces to Cerevisiae. We have long pieces of DNA with 5 genes of them, which have been clearly transferred. In this case, we know because the sequence is 100% identical to the donor, and it's not in the original strain. So we have demonstration of that. Yeast to yeast transfer can occur on long evolutionary distance. Yeah. Yeah.
So the difference strain, I'm surprised that one chromosome seems to be the one homolog seems to be protected from a circular thing to reintegrate. What's your idea?
My idea is only that this is a single-step event, so it happens on once. If it were a multiple-step event, you would expect that the 2 chromosomes will be equally affected, and it has never been like that, which suspect that it is a single-step event occurring on 1 chromosome copy and not ignoring the other chromosome copy. If they would recombine afterwards, we would see kind of intermediate. We have sporulated quite a number of them, and so far, with our analysis, we have not seen recombination between the normal and the macrotein, but we have to look for more for that. Yeah.
So the copy number of the pyrophosphatase, because during heterologous amine isolation, pyrophosphate remains attached.
Yes.
And then to remove it.
Absolutely, you are right. We have not done the biochemistry of that, so I can, absolutely.
But it would be interesting if the copy number of the pyrophosphatase increased as well, so that would allow you.
Yeah, we could look because we have the data, and I have to look at that. Yeah, we look whether the gene is overexpressed or something like that, because we have the data, but we have not done the biochemistry of the tRNA charging, which could be something.
To go back to the beginning where Saccharomyces lineage has very few introns.
Yeah.
And what I had always understood this to be that most of the introns might have been eliminated by cDNA homologous recombination methods. Do you think not?
Yeah, it's possible. There is a paper more recently they measured, because we have so many yeast, they measure the rate of loss versus gains. So over time, there is gain and loss, but loss exceeds the gains. And it is quite possible that the loss has occurred by the part of the retrogene, basically retrogene, which is quite possible mechanistically. But it has occurred also on the basal lineages, because even Yarovia, which is claimed to have more introns, that's 15% of gene split instead of 3 to 5, which is more, but it has still a low number of split genes. So it was already present in the ancestral part, probably, maybe by the same mechanism, that I don't know, but it is clearly ancestral to the budding yeast. They all have that.
Are you in a situation to determine, is there a limit to the possible size, the possible length of chromosomes? And so the older chromosomes are longer. And for Al is this story where he is artificially, but here you're not exceeding the normal limit.
Yeah, I'm going to 2.5 megabase, which is larger than the largest chromosome of Cerevisiae, not the largest chromosome of yeast, because Pombe has 4 megabase, 5 chromosomes, so it's not the largest, but it's the largest for Cerevisiae. According to what people say during budding, it should be a segregation problem, because the chromosomes are so big that in fact the budding, the end of the chromosome will be lying behind and not segregating properly, which is possible that we have mutants like that, but not frequently enough to have a significant impact on the fitness, on general fitness. You know, that's.
So,
Yeah, thank you.